All right, welcome everyone. This is lecture 2 of 6047, 6878 HST 507. I'm actually going to be giving this lecture from the airport because my connection flight is not getting back in Boston on time. So hope you'll bear with me. Start. So today we're going to be talking about sequence alignment and introducing dynamic programming. So basically both sequence alignment is a fundamental computational challenge in biology, and dynamic programming is a very foundational algorithmic technique that is going to be pervasive throughout the class. So here we are. We're starting module 1 of the class on aligning and modeling genomes. And in this module, we're going to be learning about dynamic programming today. We're going to be learning about database search, and I'm actually going to be in class in person on Thursday. And then next week, we're going to be learning about hidden markup models. So what is the challenge of alignment? The challenge of alignment basically stems from the fact that every single living organism in the planet today is thought to arise from a single common ancestor, which then gave rise to the huge complexity of life forms that are found today. And in the previous slide, I only showed primarily the diversity of mammals, but they're a tiny little part of the tree of life. You can see here the incredible complexity in life forms, in body plans, that you can see throughout the course of life. And this is the bottom half of that slide. And then humans are sitting somewhere around here in the vertebrate tree. The other thing to realize is that that previous tree only showed the species that are still alive today. And this is, again, a tiny little minority of the species that lived throughout the history of the planet. Extinction is very much part of life. This is this huge diversity of dinosaurs, for example, that roamed the earth, a tiny little branch of which became modern birds and ultimately crocodiles on another lineage, but the vast majority of which was extinct after a very big asteroid hit just north of the Yucatan Peninsula. So what do we do with this species? We can basically start building phylogenetic trees relating the species, and we're going to learn much more about phylogenetic trees later in the term. And then once you have a global alignment of at least the regions to compare, that's where the challenge of low-level sequence alignment comes in, where you can actually see here the order of all of the genes in the different species is largely preserved. So that basically means that within each of those, we can actually start aligning and lining up basically the nucleotides at high resolution. So this scales not just for a few genes, but for hundreds of genes. And that means that we can actually start building genome-wide alignment. So we can actually span the entire genome and use that to identify functional alignment. Here's one example of the usefulness of alignments. Basically, when you compare many species, you can actually see that there are very different evolutionary rates at different locations. And the slower evolutionary rate basically indicates higher conservation and higher functional significance of those regions. So if you're scanning this region, you'll basically notice that there are many different blocks of conservation. And these largely correspond to the exons, the protein coding regions of this particular gene. But then there are other regions that are not overlapping exons. So in addition to estimating mutation rates, we can actually start looking at the patterns of evolution. And we're going to be learning much more about that in the Comparative Genomics lecture later in the term. These evolutionary signatures basically allow you to now recognize protein coding regions, RNA structures, microRNAs, regulatory motifs, solely based on their patterns of change within these regions, and then systematically annotate genomes from those patterns of change. This is the first alignment. You'll see this term. This is basically looking at 4 species of yeast: Saccharomyces cerevisiae, Paradoxus, Mucatae, and Bayanus. They're wrapped around, and you can see here stars in every position where all the species share exactly the same letter, and then gaps, dashes, when one species has gained a nucleotide or when another species has lost a nucleotide. So in this region, you'll see that there are blocks of conservation, and these blocks, in fact, correspond to known binding sites or different regulatory elements within this interval of the yeast genome that actually determines which sugar will be metabolized, whether it will be galactose or glucose. And then GAL4 binds here to activate GAL1 and GAL10, which are transporters and processors of galactose. And then MIG1 binds here to basically repress the transcription of these genes when glucose is in the media, because the yeast prefer glucose. It's a much more easy to metabolize sugar. You can see the TATA binding protein binding sites here, TATA. So that means that we can actually start reading evolution to reveal functional elements. And what we need is a systematic way to actually generate these alignments. So today's goal is, how do we actually align two genes? And first, we're going to introduce sequence alignment. Then we're going to introduce the principles of dynamic programming, and then we're going to use dynamic programming for sequence alignment, and then turn to some variations of dynamic programming and more advanced topics. So genomes change over time. If you have an ancestral sequence, you can have a mutational event from C to G. You can have deletion events where the C and the A are deleted and the sequence is condensed. You can have insertion events. And what you end up with is a different sequence over time. The goal of sequence alignment is to effectively figure out what are the series of evolutionary events, what are the series of operations that transform one sequence into another sequence. And of course, defining the set of evolutionary operations is the first part of formalizing the problem. In this particular case, they're insertions, deletions, and mutations. And these are symmetric operations, which actually allow for time reversibility. This is actually part of the design choice. Therefore, when we're comparing human and mouse, instead of trying to figure out what was the common ancestor before we carry out an alignment, we can simply reverse the time direction in one of the two branches, and then think about the set of operations that transform human into mouse or mouse into human as the x plus y distance on each of the branches together. We can start defining an optimality criterion, namely what is the minimum number of operations or the minimum cost of operations. And again, it's kind of impossible to actually infer the exact series of operations because the sequence may have actually gotten further from each other and then closer from each other and then further from each other again. But what we're going to use is Occam's razor and simply find the minimum number of operations that transforms humans into mice and vice versa from all of the possible transformations. And we're going to be designing an algorithm that actually achieves that optimum or approximates it. And so basically, first we define the operations, then we define what would be optimum if we could get there. Then we're going to design an algorithm to actually get to that optimum. And then again, the tractability of the solution depends on the assumptions that we make in the formulation. So what we're going to be faced with throughout the term is a trade-off between biology and computer science. On one side, the closer we are to achieving the biological goals, the higher the relevance, the correctness, handling special cases, predictability. And on the other side, the closer we are to CS, the easier it will be to actually define algorithms for the problem, understand the assumptions, the tractability, the implementation, and the computability. And of course, not all decisions are conflicting. Sometimes we'll make a decision that will be both making the biology better and making the computation more tractable. So let's walk through these processes to basically formulate the problem of sequence alignment. So first, let's talk about the longest common substring. So given two possibly related strings, S1 and S2, what is the longest common substring, assuming that there are no gaps? In this particular case, the longest common substring is, here, for example, you see GT is a very long, is a two length two substring, but then TCA is a longer one. So if we offset by 1, then we get TCA. Can we do better? Search a little bit. You might realize that if we actually offset by -2, you can actually get GTCA to match GTCA in the two sequences. So we can actually get a length of 4. So How did you do this in your head? Basically, the easiest way to do that is to just start scanning the sequences past each other and basically look for which of the offsets defines the longest sequence. And the way to do that is to basically try every single offset. Then once you have an offset, basically start comparing every pair of nucleotides. So that's an order n algorithm because there are n possible offsets. And then for each offset, you need to compare all of the characters. Can you do better? Of course. As soon as you find a mismatch in one of the offsets, you can just stop. And that actually makes the algorithm faster. And that speed up could make it linear or still quadratic, depending, of course, on the number of mismatches between the sequences. The second formulation is to actually look at the longest common subsequence. Hold on one second. I'm actually doing a recording. Can I wait here until we board? Μπορώ να περιμένω μέχρι να κάνουμε boarding. Μόλις έχω κάνει ήδη scan το... Μπορώ να έρθω τώρα να το κάνω το pre-boarding. Μπορώ να σας δώσω το κρεβατήριό μου και θα έχουμε καθαρίσει. Έχω μόνο μία καταπλάτη. So the second more complex way of looking at this problem is to now start introducing gaps. So this is one more evolutionary operation. This brings us closer to the biology, but this takes us further from computational tractability. So now we have to come up with more sophisticated algorithms. If you're searching now for the longest common subsequence, allowing for gaps, you can basically get a length of 6 because if you basically space out this particular sequence by introducing 2 gaps, you basically end up with the A's matching, the G's matching, the T's matching, and then TCA matching at the end. You now have length 6. And this, of course, immediately implies a set of evolutionary operations. Losing insertion here, 2 deletions there, and a mutation there. In fact, exactly the same way that we saw in the earlier example. So this is, in fact, quite related to edit distance, the number of changes needed to transform sequence S1 into S2, given a uniform scoring function. And this is just, if we assume that every evolutionary operation is equally likely, and therefore that every evolutionary operation should be weighed equally, cost the same amount. So we can actually make the formulation a little more sophisticated to go a little closer to biology by basically transforming it into actually sequence alignment. So we now still allow gaps with a fixed penalty, with insertion and deletion operations, with a unit cost for each character that is inserted or deleted. But now we have varying penalties for edit operations. So transitions between pyrimidine and pyrimidine, or purine and purine, cost less, whereas transversions between purines and pyrimidines cost more. And the reason for that is that polymerase very often confuses A with G and C with T. So A and G are both purines, C and T are both pyrimidines, and they're much more easily translated into each other. So we can translate that into a scoring function that will basically have a penalty for a mismatch, which is the same whether you are an A to a G or a G to an a C to a T or a T to a C, but is higher if you change an A into a C or a T, and similarly a G to a C or a T, and vice versa. And then, of course, the matches are all the same strength. So you basically get a +1 for every match, and then -1 for transversions, and then -1/2 for transitions. These costs can be further tuned to account for the frequency of the different nucleotides, or the background rates for each of those, and so on and so forth. So basically, introducing only this concept of transitions versus transversions, we end up with a 3-parameter model that we can then fit and make a much more accurate biological alignment without actually much more of a cost computationally in terms of runtime, simply a little more sophistication on computation time. There are further formulations, so you can actually start going beyond this linear gap penalty, which costs the same for every nucleotide that you insert or delete, regardless of how many nucleotides you have already inserted or deleted. An affine gap penalty that basically has a big initial cost for starting or ending a gap. That basically, if polymerase starts skipping, then the initial cost might be large, or if a big deletion happens, the initial cost might be large, but then the size of the deletion matters little. If you insert 13 or 14 or 15 nucleotides, the incremental cost is relatively small. We could have a general gap penalty function, which basically says, give me the length of the gap, and I'll give you a cost. So that's a general gap penalty, but this no longer allows us to compute this using the same model. We could also have frame-aware gap penalties. So basically, if the insertion or deletion is a multiple of 3, then within protein coding regions, this is actually much less disruptive, enabling us to start, making alignments much more relevant to protein coding segments. And this can actually be computed using the same computational complexity as the affine gap penalty. And then beyond that, we can start seeking duplicated regions, rearrangements, etc. So what you can see here is this increasing complexity of the formulations, basically starting from simply no gaps, finding the longest common substring, then allowing gaps, then allowing varying costs for different matches or mismatches based on purine, pyrimidine transitions and transversions, and then including varying costs for the gaps. So the model we're going to look at today is affine gap penalty and how we can achieve that quite efficiently. So another thing to realize is, can we actually do that in a brute force fashion? So in order to basically search for the specific alignment that relates these two sequences, you could have basically said, well, let's simply try them all. So how many such alignments are there? The way to think about that is to basically say that the longest non-boring alignment can maximally have length n plus m, where n is the length of the first sequence and m is the length of the second sequence. So otherwise, you would basically end up having a gap aligned with a gap, and you can just simply condense the sequences by removing the two gaps. So therefore, if you think about it this way, alignment is equivalent to a problem of gap placement, where you're basically placing the gaps in each of the sequences in a way that maximizes basically the number of characters that are aligned to identical characters across the different sequences. And this is effectively a problem of choosing the n ways that one could put n plus m, n gaps into an n plus m sequence for each of the sequences. And each position, you're basically choosing a yes/no answer of whether I'm placing a character or a gap. And that basically is, approximately equivalent to an exponential number of possible placements. So at every position, I can choose to insert a gap or not. And then there are n such positions. So it's effectively a 2 to the n order problem. So what we need is a faster solution for finding the best alignment. What we need is a polynomial algorithm to basically find the best alignment among an exponential number of possible alignments. And every time you're looking for a polynomial runtime to explore an exponential search space, a technique should come to mind, which is dynamic programming. So let's now introduce the principles of dynamic programming, which will be pervasive in computational biology, and then turn and use them for sequence alignment. So how do we turn exponentials into polynomials? So let's start with a seemingly unrelated topic of Fibonacci numbers. So as you know, Fibonacci numbers start with 1, 1, and then every next number is the sum of the previous two. So 2 is the sum of 1 plus 1, 3 is the sum of 2 plus 1, 5 is the sum of 3 plus 2, 8 is the sum of 5 plus 3, 13 is the sum of 8 plus 5, 21 is the sum of 13 plus 8, and so on and so forth. So you can see here how this is in fact computing the sides of increasingly large squares. And in fact, this draws a perfect Nautilus spiral. So because of this very simple property of summing up the last two numbers, Fibonacci numbers are actually pervasive in nature. So if you look at the number of rabbits per generation, if rabbits basically take one generation to come of age, and then you start with a pair of rabbits, they have, one pair of children, and then by the next generation, they basically start, procreating. So you basically have these rabbits that are coming of age, and then they have a pair of children here, which are coming of age, and then they have a pair of children, which are coming of age, and then they have a pair of children, which are coming of age, and then have a pair of children, and so on and so forth. This basically allows you to now trace the initial set of rabbits, which are then having a new pair at every generation. So you have a new branch. And then every one of those branches, in fact, creates a Fibonacci tree under that. This is basically leading to the 1 plus 1 equals 2 plus 1 equals 3 plus 2, 3 plus 2 equals 5, and so on and so forth, which basically leads you to this growth of the Fibonacci numbers. Now, similarly, if you look at the number of leaves in many trees, you basically end up with a Fibonacci series. If you look at the spirals of a Romanesque or the Nautilus size or coneflower spirals, you actually have two different Fibonacci series here. The ordering of the leaves. So Leonardo Pisano Fibonacci was obviously born in Pisa in 1970, and he came up with this series and these examples. So you can code this up in Python very easily. You can basically say, well, let's define Fibonacci of n. If it's n equals 1 or n equals 2, you just return 1. Otherwise, you just simply return Fibonacci of n minus 1 plus Fibonacci of n minus 2. And if your goal is to compute the nth Fibonacci number, you'll basically, arrive at the right answer, and you're going to end up with this. But if you analyze the running time of this relatively simple piece of code, you basically end up with a lot of computation. So basically, to compute t of n minus 1 plus t of n minus 2. To compute t of n minus 2, it's basically t of n minus 2 plus t of n minus 3. To compute t of n minus 2, it's t of n minus 3 plus t of n minus 4. And basically, you end up with these trees that are repeatedly nesting inside each other. And that, in fact, leads to an exponential growth in their runtime. So basically, this is an extremely inefficient way of calculating Fibonacci numbers. Instead, what you could do is fill out a table where to compute Fibonacci of n, I'm actually going to solve a seemingly harder problem, which I'm going to compute all of the Fibonacci numbers between 1 and n, systematically from the bottom up. I'm going to put them in a table called Fib table. And then for I in the range of 3 to n plus 1, I'm just filling in my table. And the beauty of this is that every single time I'm filling up one entry, I have already computed the previous two entries. So instead of starting from Fibonacci of 6 and then expanding this very large tree, I actually start from Fibonacci of 0 and 1 and then fill out only one line of that tree, which basically results in a linear time algorithm. So that's actually the very principle of this bottom-up computation that we have in dynamic programming. We will always build up a table in order to have all of the solutions to the subproblems that we're going to need already pre-computed in order to then be able to use them at the next iteration. So the iterative solution basically revealed these identical subproblems. It ordered the computation to enable the reuse of the results, systematically filled in the table of the results from the bottom up, and then expressed the larger problems from their subparts. And the ordering of the computation matters. The naive top-down approach is in fact very slow because the results of the smaller problems are not available. And then that leads into a lot of repeated work. Whereas for the systematic bottom-up approach, we were able to be successful because we systematically solved every subproblem, we filled in the table of the subproblem results in order, and then we simply looked up the solutions instead of recomputing. And these are the hallmarks of dynamic programming. Basically, what you need is to have an optimal substructure so that the optimal solution to a problem instance contains within it optimal solutions to the subproblem instances. You also need to have overlapping subproblems. Basically, what we had in Fibonacci was a limited number of distinct subproblems that were repeated many, many times. And typically, we're going to be looking at optimization problems for dynamic programming. So the Fibonacci example was not an optimization problem, it was simply a computation problem. And in fact, it's a little bit of a contrived example because Fibonacci actually has a closed form solution. But it basically taught us the principles of filling out that table bottom up. Here, the optimal choice will be made locally by basically thinking the maximum of a sub-solution score and then optimizing by making that choice. And then the score will typically be added through the search space, and traceback will be the last part, where after we have computed the score of the optimal solution, we're going to be tracing back through our saved solutions to find the optimal path. For Fibonacci, there was no such optimality. There were no choices. You should also realize that dynamic programming is actually middle of the road in terms of that range of difficulty. There are easier problems where greedy choice is actually possible at every step. Dynamic programming requires a traceback to find the optimal path. And then there are much, much harder problems, of course, where no optimal substructure is possible. For example, if there are dependencies between the subproblems, you can't use dynamic programming. So how do we set up dynamic programming? Well, basically, first of all, we need to figure out how to parameterize a matrix of parameterization. In this particular case, which is simply a one-dimensional table. Make sure that the subproblem space is finite, not exponential. Choose how to traverse the matrix so that the sub results are already ready when you need them. And then define the recursion formula. In Fibonacci, it was simply figuring out the function of the sum of the parts, which was a plus operation. And then for optimization problems, we're going to have to remember the choices. And typically, that function will require some kind of minimum or maximum, or basically will give you a choice among multiple options. And you'll have to figure out which of these options to use. And once you have all that, you can just start computing. Fill up the matrix systematically, find the optimal score, and then trace back from that optimal score to find the optimal solution. So now let's use these principles of dynamic programming for sequence alignment. Figure out how we can express this sequence alignment problem using an additive score, build up a solution from the smaller parts, and then start computing. So how do we apply dynamic programming to sequence alignment? So the key insight here is that the score is actually additive. And you can express larger problems as functions of smaller problems. So if I have an alignment of the sequence S1 and the sequence S2, part of that larger problem of aligning the entire sequences is actually aligning the prefixes of S1 to the prefixes of S2. And if I start figuring out the optimal alignment of prefixes, I can then make the prefixes larger by using the prefixes that I have already computed, and similarly extend, this I and J index of how big of a prefix I'm looking at, all the way until I equals n and j equals n. So we're going to be computing the best alignment recursively. So for a given aligned pair, ij, the best alignment will be the best alignment of S1 all the way to I and S2 all the way to j, and the best alignment of S1 all the way to the end. And we're going to be doing this by adding only one character at a time. To prove that this must be the case, we're going to be using a cut and paste argument. And this is something that you will see in 646 and even in 606, where you basically say, if the best alignment here was not made up of best alignments within here, then I could simply take a better alignment for any of them and then construct an even better alignment than the best alignment, which of course is impossible. So therefore, in order to have a best alignment for the top, I must have had best alignments in the bottom. So the second key insight is that we're going to be computing the score recursively. Instead of sort of starting from the top and then chopping up the sequence in a bunch of random ways, I'm going to be computing systematically all of the prefix alignment scores for increasing I and J for each of the two sequences. So instead of basically having to recompute CGTTG with two different sub-problems ending up here, I'm going to be computing from the bottom up and growing the size of the prefixes. So there's two solutions for doing that. One is memoization, where you basically create a big dictionary indexed by the aligned sequences. And whenever you encounter a new pair of sequences, you basically say, hey, is it already in the dictionary? If it is, then I just look up the solution. If it's not in the dictionary, then I compute the solution, and then I insert the solution in the dictionary. So in this particular case, I could have simply said, CGTTG. This is, I'm just going to hash this into a key, and then I'm going to look up every single time whether the key for CGTTG already exists in my set of solutions that I've already computed. And if it doesn't, then I'm going to compute it and add this key into the dictionary. And if it's not already there, then I'm going to be and if it is already there, then I'm just going to simply look it up. So that's memoization, basically ensuring that there's no duplicated work, where you only need to compute each sub-alignment one. And this is kind of like a top-down approach, where you don't have to worry about which sub-problems we're going to be solving. You're going to start at the top, and then whatever sub-problems you need to compute, you compute, and only those that you actually need. The alternative is a bottom-up approach, where you basically have a very big table, just like Fibonacci, indexed by these prefixes, I and J, and you fill in that table from the beginning all the way to the end. So you know that you already need every subpart, and then you just fill up that table with every subproblem. This is actually guaranteed to explore the entire search space. So memoization can actually be more efficient if in the end you only explore a very small subset of the space. But this is actually much more efficient if you do explore the entire space, because you know the order of computation. You can actually order things much more efficiently. And this ensures that there's no duplicated work, because you only need to compute each sub-alignment exactly once. And it's very simple computation. So this is the approach that we're going to be using today. So this is the last insight, that the optimal prefix alignment score can be computed in a single entry of that matrix. So I can basically compute a matrix where for every position ij, I simply compute the score of aligning the ith prefix of S1 to the jth prefix of S2. And then that score, the maximum score that I could possibly obtain by aligning those two sequences, will be stored here. And that will allow me, when I'm computing the next entry, to basically choose whether I want to move from here, from here, or from here, which would correspond to a gap in this direction, a gap in that direction, or a mesh in that direction. So that basically means that if I align every character to every character in order with no gaps, I'm going to be moving along the diagonal. And every single time I want to add gaps in S1, I'm going to be moving this way, basically including, sorry, gaps in S2, including more characters from S1, but no characters from S2. And then moving that way, including characters from S2, but no characters in S1. That corresponds to a gap in S1. And then every time there's a match, I move diagonally. Okay. So now the path through this matrix is in fact corresponding to an alignment. If I want to insert gap, 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 match, match, mismatch, gap, 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 mismatch, and so on and so forth, I can basically traverse this matrix, giving a unique alignment between S1 and S2 for every path through this matrix. And there is an exponential number of paths through the matrix, just like there's an exponential number of alignments. But I can actually compute the optimal score of going through this matrix across any path using dynamic programming, as we will see. So that's the last key insight, that every optimal alignment gives me a path through the matrix. So the best alignment is simply the best path through that matrix. So here's the alignment that we had seen earlier, where there's an insertion, a deletion, a mismatch, and another deletion. And you can see here how I can represent this as a path through the matrix by having a gap in S1 with a character in S2, a match, a gap in S2 with a character in S1, a match, a mismatch, where I'm including a character from each sequence, but they're mismatching, so I'm paying a penalty of a mismatch, and then an insertion in the S2 sequence, an insertion in the S1 sequence, the A corresponding to a gap in the S2 sequence, and then a match TCA. Okay. So this is the key set of steps before we're now able to just go through and apply dynamic programming. So you can hopefully see now that the dynamic programming approach is iteratively growing the best alignment solution and basically walking through all of the characters of increasingly long prefixes of the two sequences. So we're going to be computing all of the alignment scores from the bottom up, defining a matrix indexed by I and J of all of the prefix alignment scores of S1 and S2, all of the optimal prefix alignment scores. And then we're going to be filling up this table recursively from smaller to bigger alignments, so iteratively from the bottom up. We're going to be expressing the alignment of the I plus first prefix of S1 and the J plus first prefix of S2 by one of three possibilities. Either we're going to be extending the alignment from Mij, or extending the alignment from M-1J, or IJ-1. And for each case, we're only doing a local computation, which takes order of 1 time. So we're going to be computing these alignments recursively by only using local rules and only looking at the neighboring cells. To compute the next alignment based on the previous alignment, just like the Fibonacci numbers, we're adding two numbers that were already in the table. Notice these are not parentheses of calling a function. These are just brackets of looking up an entry. And then the table lookup avoids any repeated computation. So we're now going to be computing the score of a cell from smaller neighbors, where the maximum score of aligning the ith prefix of S1 to the jth prefix of S2 is in fact simply the maximum of the I-1 to J alignment. And that's the maximum possible score I could have, minus the score of a gap and the cost of a gap. And then the second possibility is that I'm actually going through and adding both a character from each of the sequences, and then the score corresponding to that character that I just added. And this could be either a match or a mismatch, or basically, choosing to include a gap in the other sequence. So there's only three possibilities for extending by one nucleotide: a gap in one species, a gap in the other species, or a match / mismatch. So to compute the scores for the prefixes of increasing length, we just start with prefixes of length 1 and extend by 1 each time until all prefixes have been computed. And when you reach the bottom right of your matrix, the alignment score between S1 prefix all the way to M and S2 prefix all the way to N is the alignment of the full S1 sequence to the full S2 sequence. And then we can simply trace back to construct the optimal path. So let's go through the 5 steps of the dynamic programming solution. We basically have to find the matrix parameterization, and we did that through this prefix length. Make sure that the subproblem space is finite. And again, it's just an n-squared subspace. It's just that matrix that we have to traverse in terms of path. Choose a traversal order, and then here it's important to either to always make sure that the top and left entries are already available, and this can be achieved by computing all the columns, computing all the rows, computing diagonally. So the recursion formula, gosh, it's going to be a little awkward.